So let's dive a little deeper into the metabolic response and how it follows neural activity, and importantly, how does it give rise to the bold signal? Now, typically, if you were to look at the saturation of oxygen inside the blood vessels in the vascular system, studies that have looked at this, on average, report that in the arterial side, saturation is somewhere above 90% saturation of oxygen. Once you get to the capillaries, which obviously are half arterial and half venous, the oxygen saturation tends to be closer to 80%. And then once you get to the venous side, the saturation of oxygen falls down to 60%, meaning now there's 60% oxygenated hemoglobin and 40% deoxygenated hemoglobin. Now, studies that have looked at how these numbers change with neural activity have reported that generally the cerebral blood flow and the cerebral metabolic rate of glucose, so the rate at which glucose is metabolized out of the vascular system, is just about 50%. So with activity, the amount of flow of blood through the vascular system increases, and somewhere around 50%. Of course, this is an average. And the same is true for glucose. So the metabolism, the rate at which glucose gets used, the cerebral metabolic rate of glucose, also increases by just about 50%. What is somewhat odd, and we will spend some time discussing this, is that whereas CBF and CMR glucose increased by 50%, the metabolic rate of oxygen, so the rate at which oxygen is metabolized during activity, only increases by 5%. So in a sense, there's a dissociation between the CBF and CMR glucose, which increased by 50%, and the CMR oxygen, the cerebral metabolic rate of oxygen, which somehow only increases by 5%. Now, overall, when people have measured the levels of saturation of oxygen in the vascular system, they tend to report that on the arterial side, during activity, so in the area proximal to the stimulation or to the neurons that became active, the saturation of oxygen inside in the arterial side increases maybe by 3%. On the other hand, at the capillaries, the saturation of oxygen seems to increase by just about 5%. Finally, on the venous side, there is the largest increase, with the increase going up by just about 12%. So going from 60 to 72%. So it goes up by 12 percentage points, I should say. Now, of course, the question is, why does this happen? And in particular, why does CBF go up so much, but the rate of oxygen use only goes up by 5%? A number of different theories have been proposed as to why there's such a change in the cerebral blood flow, 50%, for only such a tiny change in the cerebral metabolic rate of oxygen, just about 5%. The first idea came from Fox and colleagues, and actually Fox and colleagues are those who derived the measurements that I showed in the previous slide. So they observed for the first time this big dissociation between CBF and CMR02 with activation, and they thought that maybe the two dissociate, the cerebral flow and the use of oxygen dissociate, probably because, this is their speculation, oxidative metabolism, so aerobic metabolism, might already be at ceiling during rest. So in other words, they thought that perhaps just at rest, so with no extra activation, the oxidative metabolism or the aerobic metabolism might just be maxed out already, somewhere close to 99%. So whenever a neuron would suddenly start bursting with activity and require more ATP to support its function, this additional energetic requirement might be taken care of with an increase in the faster, although less efficient, anaerobic metabolism, so really through glycolysis. So the idea is oxidative metabolism, aerobic metabolism, is just going on the whole time. And whenever a neuron requires more ATP, so requires more energy, that is addressed with an increase in glycolysis. So the use of glucose transformed into 2 molecules of ATP and pyruvate and then lactate. Indeed, we do know that lactate, which is a byproduct of glycolysis, does increase with neural activity. So this dissociation between CBF and CMRO2 might be due to the fact that CBF, even though it's carrying both oxygen and glucose, it's really just only the glucose part of it gets used for extra energy through glycolysis. The oxygen itself doesn't help, because this was the hypothesis of Fox and colleagues, because the oxidative metabolism might already be maxed out at baseline. So this was their theory. Now, a different point of view was proposed before that by Malonic and Greenwald. Now, Malonic and Greenwald, they thought that the difference had to do with a difference in how local or coarse the control of metabolism can be. So here's what they did. They used a cat model, and they implanted in the cat a near-infrared spectroscopy detector. So this is just a different form of imaging, and it uses infrared light in order to detect concentrations of oxygenated hemoglobin and deoxygenated hemoglobin based on the fact that the two reflect differently light. So you can shine light inside the brain, and the reflection according to the concentration of oxy and deoxyhemoglobin will be different. So you can tell what's the concentration of oxy and what's the concentration of deoxyhemoglobin. And so this was the preparation, and then they just simply showed a visual stimulus to the cat. So for example, these vertical bars. And then they observed the changes in concentration of oxy and deoxyhemoglobin in visual cortex. And what they saw was that deoxyhemoglobin, the graph on the left, so this is time, and this is change in concentration from baseline. Now, deoxyhemoglobin, and the brown area is this gray area, sorry, is the period during which stimulation was taking place. So what they observed is that deoxyhemoglobin had a quick spike, and it makes sense because any oxygen lying around might be used for metabolism, but then it would fall very quickly and then slowly go back to baseline. On the other hand, the concentration of oxyhemoglobin would very slowly build over time, peak after 5 to 6 seconds, and then fall off again. And on top of this, they also noticed that this change in deoxyhemoglobin, this increase in deoxyhemoglobin, was very local in nature, only happened nearby the neurons that were actually active, nearby visual cortex, and the specific neurons that process the stimulus that they were showing the cat. Whereas oxyhemoglobin, this increase in oxyhemoglobin appeared to be much broader space-wise. So this reaction appeared to take place in a broader area compared to where the active neurons were. And so their thought was that CMR2, the use of oxygen, might be controlled at a finer scale, might really only happen in those 5 neurons that are active, let's say, whereas CBF is a more systemic response. And so if you need to increase CBF, you just need to increase CBF sort of at a larger scale. You can't only increase CBF in, the 1 or 2 capillaries where the neurons that are active are. And this difference in control is what leads to a 50% increase of CBF for only a 5% increase in CMR02, or the way they very poetically said in their paper, you're really watering the garden for the sake of one thirsty neuron. Now, the third theory, which I also think is the one that most people tend to believe today, is known as the oxygen limitation model. And the main proponent of it is Buxton. Now, the idea is the following. The idea is that the large increase in CBF, so the 50% increase in CBF, is necessary, is needed in order to get a 5% change in the oxygen metabolism. So the two are not dissociated, are not decoupled, the way that Fox and colleagues said. It's just that in order to get that little 5% increase in oxygen metabolism, you need a 50% increase of cerebral blood flow. And the idea is the following. At rest, how can you calculate the rate, what is the rate of oxygen metabolism at rest? Well, it's a function of the concentration of oxygen in the arteries. For the sake of this example, I'm going to say that that's constant, okay? Then it's a function of the cerebral blood flow, which means just how many oxygens are floating by bound to hemoglobin. And the so-called extraction fraction, the percentage of oxygens that you pull out from the cerebral blood flow. Now, we do know that at activation, the cerebral blood flow goes up by 50%. However, CMR02 only goes up by 5%. So algebraically, the only thing that could possibly be happening is that the extraction fraction is going down, which means as a proportion of the number of hemoglobins and oxygens that are floating by, at activation, even though there are more floating by, as a proportion of that more, you're pulling out fewer than before. Maybe you were pulling out 20% of, oxygens at rest, and now you're only pulling out 14 or 15% oxygens. Okay, so CBF goes up, so now there are more oxygens available. Proportionally, you're pulling out fewer than before. Now, if you pull out fewer oxygens than before, what happens to those oxygens? Well, as a consequence of this decrease in the extraction fraction, all those oxygens that have increased because the CBF went up will now end up in the venous side of the blood vessel, which means there will be a flooding of oxygen inside the venous side. And this, of course, is the whole basis of BOLD, right? BOLD is a consequence after neural activation. There's a change in the metabolic, there's a metabolic response. The consequence of that metabolic response is an increase in CBF, an increase in cerebral blood volume. The net result of that is what you see here. There's a giant increase in the amount of oxygen that is inside the veins. And you remember from the graph that I used at the beginning of this lecture and the previous one, as we increase the proportion of oxygen in the blood vessel, the magnetic field becomes smoother. As the magnetic field becomes smoother, protons resonate at the same Larmor frequency. If they resonate at the same Larmor frequency, it'll take longer for protons to dephase, so the T2 star signal will be stronger. So the way in which BOLD happens to emerge from neural activation is that at activation, we get this massive increase in cerebral blood flow. So more oxygens are going to a certain part of the brain. Proportionally, we pull out fewer oxygens of those that are floating by. So we end up with many more oxygens on the other side, that is to say, in the vein. As we get this proportional increase of oxygenated hemoglobin in the veins compared to deoxygenated, the magnetic field becomes smoother and we get the BOLD effect. Now, the question, of course, is why is there this increase in why do you need this increase of this flooding of oxygen in the veins? Well, the reason is the whole basis of the oxygen limitation model. And the idea is that when oxygens are floating by inside a capillary, they have two choices. Of course, it's passive choices. They can either keep floating down into the vein, or they can float out into the mitochondria. And both of these processes are passive. Okay, it's not that any oxygen decides where to go. It's a passive process. Now, we want more oxygens to go out of the bloodstream into the mitochondria. So one way to do that is to increase the cerebral blood flow, pack as much oxygen as possible in the vein, and now by osmosis, the next oxygen will not want to go down into the vein because there's already so much oxygen there. It will want to float out. In other words, by flooding the veins with oxygen, you're increasing the oxygen pressure inside the venous side. As that pressure grows, the next oxygen will want to float out into the mitochondria instead of float down into the vein. So this is what Buxton thought is the link between the increase in CBF, which increases massively the presence of oxygen in the vein, and the increase in CMRO2, or the increase in number of oxygens that float out into the mitochondria. So according to Buxton, this flooding is a functional role. It increases the pressure of oxygen in the venous side, and this increase leads to a greater diffusion across the membrane into the mitochondria of oxygen. So the whole idea of this model is that the vascular system relaxes the smooth muscle around capillaries. This leads to a giant increase in the amount of oxygens that float by. As more and more oxygen floats by and accumulates in the venous side, this will incentivize a couple more molecules of oxygen to float out into the mitochondria instead of down into the veins. So there's a functional link between you need to increase by 50% the amount of oxygen floating by in order to convince 5% more oxygens to float out into the mitochondria rather than to keep floating down the blood vessel. So the link between the 50% increase in CMRO2 is functional. You need a 50% increase in CBF in order to get a 5% increase in CMRO2. Let me show this with a graph and with some numbers, okay? So what I'm showing here is up here, let's just imagine that this is the arterial side of a blood vessel, there's a capillary right here, and then there's the vein. And each of these little circles is an oxygenated hemoglobin. For the sakes of the example, I'll just assume that the oxygenation in the artery is 100%. If you remember, we said it's somewhere closer to 90 to 93%, but it just makes it easier to imagine that every single hemoglobin is oxygenated in the arterial side. So right now we have 16 molecules of oxyhemoglobin in here. We know that the extraction rate is at rest is somewhere around 20%. So this means that as these protons, as these oxygenated hemoglobin molecules float by, some 20% of oxygens will float out into the mitochondria. Okay, so in our case, given these numbers, just about 3.2. All right, and of course, the 0.2 is an approximation because of the numbers I'm using. By the way, I'm putting up here the exact same table, but using 100 as the incoming number of CBF, okay, just to make sort of the numbers a little easier to digest. So here we are, we have 16 molecules of oxygenated hemoglobin, which means our CBF is 16. The extraction rate is typically 20%, so just about 3.2, and again, the 0.2 is just for the sakes of the example, float out into the mitochondria, which means that in the vein, we now have 12.8 oxygenated hemoglobin and 3.2 deoxygenated hemoglobin. Okay, so in other words, these oxygenated hemoglobin are exerting pressure. Okay. Now, let's see what happens with activation. So with activation, we know that the CBF goes up by 50%. So if here we had 16 oxygenated hemoglobins, now down here we have 24. CBF went up by 50%. Then what happens? Well, we said that CMRO2, the cerebral metabolic rate of oxygen, changes only by 5%. So really now we're extracting a little bit more, 3.36. And again, the 0.36 is just an approximation given the numbers that I'm using. So now there's a few more oxygens trickling out. Right? So as I said before, the extraction rate, so the number of oxygens that are floating out, given how many oxygens are floating by, is going down from 20% up here, 3.2 of 16, versus 14%, which is 3.8, or sorry, 3.64 of 24. So in the venous side, right, the extraction rate goes down. So in the venous side, now we have 20.64 molecules of oxygenated hemoglobin and 3.36 molecules of deoxygenated hemoglobin. And see, now there's much more oxygenated hemoglobin in the vein than there was before. So it's the fact that there's the cerebral blood flow increases, we start packing the venous side with more oxygen. As a result of that, a few more oxygens will trickle out. Okay, so the series of events is that CBF goes up, the CMRO2 doesn't change for now. As the vein sort of gets flooded with oxygen, now there's more oxygen in the vein, so the next molecule of oxygen will have a greater tendency to diffuse out rather than to keep going straight. And that is what leads to the slight increase, the 5% increase in CMRO2. Okay, so the link here is that the CBF increases by 50% so that it can pack oxygen in the venous side of the blood vessel. And as it packs oxygen in there, now a couple more oxygens will diffuse out towards the mitochondria, as opposed to keep floating down the venous side of the blood vessel. Okay, so the 50% increase in oxygenation is needed in order to persuade 5% more oxygenated, 5% more molecules of oxygen to float out into the mitochondria.